Hello and welcome to this PIR live event webinar brought to you in part by Partners in Research Canada. My name is Ben Hobbs and I will be your host today. Today's guest is Dr. Amma Allen-Vercoe. Dr. Vercoe is a professor in the Department of Molecular and Cellular Biology at Guelph University and the recipient of the 2018 Partners in Research Biomedical Ambassador Award. Thanks for joining us, Emma. Let's take it from here.
Perfect. All right. Well, thank you for inviting me to do this. Hi to everyone there in the classrooms. So my name is Amma Allen-Vercoe, and I'm a professor here at the University of Guelph, which is in Ontario. And I work on a particularly disgusting part of the human body. I work on human poo. But actually, that's a really important part of the human body because it actually turns out to be where most of the microbes that live on the human body actually live. And it turns out that those microbes are actually very, very important. So I thought today what I'd do is rather than just give you a presentation with PowerPoint slides, is actually show you around my lab a little bit so you can see what I do actually in the lab. And so here I am today in what we call the RoboGut lab. We also call it the Poopy lab because we do a lot of stuff with poop in here, so it is a bit gross, but it's okay. You don't have to smell it. I can smell it, but you don't have to smell it. You just have to imagine. All right, so just behind me here, this piece of apparatus, I'm going to see if I can sort of show it a bit more for you here. This is the RoboGut. So I don't know whether you expected this to look like a robot of some kind, but actually it really doesn't look like a robot at all. It's actually a bunch of glassware and stainless steel, and what it is it's designed to mimic the conditions inside the human gut. And in this case, inside the human colon, which is the last part of the human gut, what we call the large intestine, where a lot of digestion takes place once you've eaten your food. And particularly the microbes in the colon, they feast on stuff called fiber, which comes from fruits and vegetables most of all. And when they do that, they make a lot of really, really good molecules to help your body. And those molecules tend to smell pretty bad. That's what makes poop smell. But smelly poop means you're actually quite healthy. So that's quite an interesting thing to think about. So here's the RoboGut here. And I'm just trying to see if you can see here. But this is the actual vessel part here. So inside this glass vessel here, that's where the microbes are actually living and breathing. And you can't see them because microbes are tiny. These are mostly bacteria. Bacteria are single-celled individuals. You can fit about 1 million of them on the head of a pin. So you can imagine you can't see them with the naked eye. But what you can see is that they kind of make like a soup in this vessel here. So you can't see through the liquid. You can see that they're inside here because it kind of makes it into a thick soup. And what we're doing is we're mimicking all the conditions that you'd find in the human gut. So for one thing, it's pretty warm in here. This black part here is actually like a jacket that fits around the vessel and warms it up to body temperature, 37 degrees C. And then the whole system, even though it looks very complicated, lots of things coming in and out, is designed to mimic those conditions. So we have lots of pumps of things coming in and out. We have this thing here coming into the system is actually what we call a pH probe. So that's actually looking to see if it's too acidic or too alkaline in this gut. And our actual colons are at a neutral pH, so a pH of about 7. And so a computer system, which this is all connected to, is making sure that a little bit of acid or a little bit of base is dripped in at just the right amount of, just the right consistency to make sure that this stays at a neutral pH. And then if I just swivel this around here, you can see this bottle here that's stirring away on this stir plate here. This, although it doesn't look like very much, that is the food that we feed the microbes. So this actually, although it looks pretty disgusting, is representative of a meal that someone has eaten and has been digested by the top part of their gut, the small intestine. And so what's left over is this kind of gross mixture of mucus and sort of food substrates that are things like lots of fiber in there, lots of starches, and it doesn't smell pretty good. It doesn't look very nice, but the microbes love it. It's like their favorite food. And so what the computer is doing, you can see this long piece of tubing here, the computer is making the media come out of here. The food is coming out of here at a rate that is needed for this vessel here for these microbes. So when the microbes ask for food, the computer gives them a little bit of food, just drips it in, so it doesn't flood it completely, so that, because that would make it feel pretty sick. But it just drips it in at just the right rate. And then the other thing that's really cool about this system is that, did you know that the microbes that live in your colon are very, very sensitive to oxygen? And that's a weird thing to consider, because, you know, you and I need oxygen to breathe. That's how we live. But these microbes actually can't cope with very much oxygen at all. And in fact, some of them can't cope with any at all. And that means that if you bring them out onto the bench and you sort of try and grow them outside in the fresh air that you and I breathe, they'll die. And so what we do here is we actually pump in some nitrogen gas, and we have a whole bunch of canisters on the wall over there. We're just pumping in nitrogen gas to keep the oxygen out. And so your body does that for you. Your colon actually does that very well for you by keeping oxygen out. I think a lot of people are very surprised to hear that there's part of their body that has no oxygen at all, and that's your colon. It's quite a remarkable piece of engineering. Okay, let's see what else I can show you. Here at the back, you see just there, let me see, this waste comes out. So actually these microbes are making a lot of waste as well, because they, just like you do, they poo too, and they poop molecules. And so we actually collect that media waste, and we look into it, and we measure it, and we look at the molecules that are being produced. And that's a way of telling us what the microbes are doing, because it turns out that these microbes actually make conversations with each other, and they do that using molecules. Instead of language like we do, like words, they use chemicals. And so we can actually decipher the language, what the microbes are saying to each other, by looking at some of these molecules and trying to figure out what it is actually that they're saying. So I'm just going to walk us briefly over here, and I'm hoping that this will work. I know that it's going to look blurry for a second, so just bear with me. This instrument here, this is, we call it a bug box, but this is actually an anaerobe chamber. So this is actually where we can grow microbes on plates, and we can do that because this whole system inside is free of oxygen. There's no oxygen in here. So we can put our arms in these sleeves, and we can manipulate microbes in the chamber, and it's all nice and warm in here as well, so the microbes are very happy. And those microbes will stay what we call anaerobic, so free of oxygen. And I don't know if you can see too well inside there, but you can see lots of stacks of Petri dishes and racks of tubes, and that's the microbes actually growing on these plates and in these tubes, in the liquid that we give them, and which is pretty incredible. So we can actually see the microbes when we grow them on a plate, because there's so many of them then on the plates. And so that's a way for us to try to see what they are and to manipulate them and to do some fun stuff with them. Okay, so let me head over to here, and I'll show you, this is another anaerobe chamber. And I think you can see inside here, we've got lots and lots of bottles of stuff. And the reason that we have lots of bottles of stuff is this is more food for microbes. And this is another anaerobe chamber, but we have to keep the food anaerobic. We can't put any oxygen in the food, so we have to store all the food in this chamber and take all the oxygen away to help us feed these microbes. Okay, so I'm going to, let's see what the time is. I'm going to stop there, and let's see if there's any questions, and I'll be more than happy to answer lots of questions, or if you want me to show you around a bit more or focus on something a bit better, then I can do that as well.
Well, thank you very much for that your tour, or sorry, your laboratory. So our first question. Oh, sorry, I'm just giving you some feedback. Is that coming from your computer, Emma?
It might do. Do you want to mute it for a second? Or
I think we're all right now. It's just stopped. Thank you. So our first question today is coming in from Maymon in Mississauga, and they're wondering, do microbes need a host to survive?
Oh, that's a really good question. And the answer is, it depends on the microbe. So there are millions upon millions of different species of microbes, bacteria. Now, the ones that live in the gut, the ones that we're talking about living in the gut, they don't need us to survive. We can grow them on plates without, and in the plates in the chamber that I just showed you, and there's no person in the chamber with them. But they do need a very specific set of conditions that if they don't get those conditions, they will die. So when you poop, unfortunately, most of the microbes in your poop will die.
So this is maybe a question that we should have started out with. Mrs. Stanley's classroom is wondering, what is a microbe?
What is, well, okay, so a microbe is the term that we use for any creature that you can't really see with the naked eye, that you have to use an instrument called a microscope to really get a good look at it. And so if you really look at the tip of your finger and you squint, and you won't actually be able to see any bacteria on there, but there will be millions of bacteria on the end of your finger. And so they're very small, you can't see them. So in the case of what I was just talking to you about, the microbes that live in the human gut, for the most part, those are what we call bacteria. And bacteria are single-celled, which means they're just floating around as single-celled entities. And to compare that to us, we're made up of billions of cells. Humans are made up of billions of cells. These are single cells, and they have very simple lifestyles. When they divide, they multiply. So that's an unusual situation. So when they, in order to become more than one bacteria, they're actually divided in half. So the only things that can divide to multiply. But they are very small and very abundant everywhere you look.
So how does the size of a microbe compare to, per se, the size of a human cell?
Well, so a human cell is probably about 100 to 1,000 times larger than a bacteria cell. So, and you can't really see too many human cells with the naked eye either. So you really do need a microscope even to see a human cell. So you can imagine how tiny the bacteria are.
Very interesting. So our next question comes in from Chantal, and she is wondering, how long does it take for our food to digest?
Oh, well, again, it depends on you as a person, because everyone is a bit different. But normally it would take about 30 minutes to an hour for your food to go through your stomach. That's the first part. It's up here. And then through the small intestine, it takes another maybe 6 to 8 hours. And then in the colon, the last part, this part that we're modeling here, it takes about 24 hours.
Okay, interesting. So Neil from Mrs. Robinson's class, he is wondering, what do you mean by microbes have their own language?
Well, oh, that's something that we study in the lab quite a bit. We're very excited about that. What we mean is that we didn't realize until really not that long ago that microbes can talk to each other. Bacteria can talk to each other. Because we thought that bacteria, because they were just single cells, that they're too simple, that they didn't do anything. They just kind of floated around and multiplied, and that's about it. Now we know that they're very sophisticated little machines, really. And in order to understand their environment, they have to be able to interact with their environment. Now, you and I do that very easily because we have our 5 senses. We can see, we can feel, we can smell, we can touch, etc. But bacteria, they don't have a nose or eyes or mouth or anything like that. So how do they do it? And it turns out that they use chemicals, and they have receptors for those chemicals on their surfaces, so that's their senses. That's how that they can sense their environment. And they can only really sense their environment by looking at the chemicals that are in their environment. And so they have very sophisticated mechanisms of looking at those particular chemicals. And that's the language that we're talking about. Because it turns out as well that as well as looking at chemicals, bacteria can also make chemicals. And they can use those chemicals to communicate with other bacteria.
You mentioned several times that you were growing microbes in anaerobic environments. How do you get the oxygen out of those chambers?
Well, you can probably hear there's a lot of background noise in this lab. I don't know if you've noticed, but a lot of that is actually from a vacuum pump. So it's a pump which literally pumps and forces the air out of a space. And then we have gas chambers or gas canisters that basically pump the gas that we want into those chambers to replace the oxygen that's being pumped out. And those canisters contain nitrogen and carbon dioxide and hydrogen, so 3 gases that we can mix them together. And microbes actually like those 3 gases quite a lot, and it just keeps the oxygen out.
Interesting. Miss Sampson's class is wondering, are there things that are even smaller than a microbe?
Oh, that's a good question, and the answer is yes, there are. And I suspect you've heard of them too, viruses. Viruses are even smaller than microbes. A lot of viruses actually infect bacteria themselves. So it's not just us that get sick from infections with viruses, but bacteria do as well. So if the bacteria are as small as I say, you have to imagine that the bacteriophage, that's the viruses that infect the bacteria, are about 100 times smaller than that. So they're quite incredibly tiny. And viruses are also a type of microbe. So there are people that study viruses, we call them virologists. And we, in fact, in my lab, we're studying the viruses that live in the human colon as well.
Okay, thank you. So Ms. Stanley's classroom is saying that you talked about the pH being alkaline. Could you maybe elaborate on that a little bit further?
Sure. So actually the pH in your gut and your colon is neutral. So on a scale of 14, the pH goes from 0 to 14. So anything that's less than 7 is acidic, and anything that's more than 7 is alkaline. Okay, so something like acetic acid, so vinegar, would be acidic, so have a low pH, and something like bicarbonate of soda or something like ammonia would be a much more alkaline product. And so the job for us in our bodies is to keep our bodies at a fairly regular pH. The only exception to that is really the stomach, which has a very low pH because we have a lot of hydrochloric acid in there to help digest our food. But in the colon, the pH is neutral. And so we have to make sure that the conditions inside the robogut here are neutral so that the microbes are happy. And normally our body does that for us. The reason we have to do that is because those molecules that I told you those microbes are making all the time as part of their language, quite a few of them are acids. And so that's the reason that we have to try and maintain this pH.
Okay, thank you. Our next question is coming in from Jacob, and he is wondering what would happen if all of a sudden all the microbes in our body were gone?
Oh, well, we kind of know the answer to that, not because of what we can do with humans, but of what we can do with animals. So there are some animals, so mice in particular, that you can raise them in a situation where they have no microbes at all living on their bodies. Okay, so not in their guts, not on their skin, nowhere. They're very difficult to raise because you have to raise them in these kinds of chambers, obviously with some oxygen in them. But they have to be very carefully maintained apart from the rest of the world that's covered in microbes. Now, the interesting thing about these mice is they're not very normal. So they don't do very well. They have to feed, you have to give them about 30 to 40% more calories than a normal mouse just to survive, because their bodies have no microbes to help them digest the food that they eat. And so that makes that quite difficult. Intriguingly, they also don't reproduce very well. So they don't have baby mice very easily. Often they require a lot of help to do that. And they also, even more intriguingly, they also have some very strange behaviors. So they behave in a very unusual way. They show less of a response to stimuli that would normally give them fear or anxiety. They don't seem to be as worried about that. It's a very interesting sort of thing to look at when you look at these animals. They really don't look very normal. But we don't have any humans that live in a germ-free lifestyle, so we don't really know what that would do to a human.
So when we're born, do we have these microbes already within us, or is this something that happens after we're born?
Oh, well, that's another great question. It actually happens after you're born. So when you're a baby in the womb, in your mother's womb, in your mother's tummy, you're actually sterile. So that's one time in your life when there shouldn't be any microbes living on you or in you. And then what happens is towards the end of pregnancy, just about when you're getting ready to be born, your body or your mother's body sort of tries to educate your own body, your little baby body, how to deal with microbes. And then at the moment of birth, when you start being pushed out of your mum, you pick up a lot of the microbes actually from your mum. So most of the microbes that come onto the human body in a newborn infant actually are coming from the mother. And then over time, the microbiome develops because you're picking up a lot of microbes from your father and from the environment in which you live, from your pet dog and all those kinds of things as well.
Very interesting. So if you're sick and you vomit, number one, would you lose a lot of microbes? And number two, would this have any effect on your overall health by losing those microbes?
Okay, so the good news is probably not. So it's not very nice to vomit. It's not very nice to have diarrhea either, which can also be a consequence of getting sick with some food poisoning or something. But the good news is that your microbes recover very nicely. So you might have a few days where you feel very low, you're still getting over your infection, but eventually your microbes will pick up exactly where they left off, and it's quite remarkable how resilient that they are.
Sophie is wondering, when you grow your microbes, do you take the samples from humans?
Yes, we do, and so it's a lot to process. It's a lot to think about, but actually, we need very fresh poop, and so we have some very brave donors who come in, and we have some private bathrooms just down the hall, and we ask them to poop into a pot for us, and then we put that into the RoboGut behind us. So it's pretty gross to think about, but we're used to it. We work with poop every day.
Earlier, you mentioned how we live in sort of a symbiotic relationship with these microbes in our bodies. Could you give an example of a chemical that they produce that's beneficial for us?
Yes, so I think the best example of a chemical that these microbes produce that's good for us is, well, actually there's a few, and they belong to a class of molecules that we call short-chain fatty acids. Okay, so you don't need to worry too much about what that means, but they have a particular structure. And one of these compounds is called butyrate. Now, butyrate actually, if you have it in the lab and just have it in a bottle from a chemical company, it's just a colorless liquid, and it has a terrible, terrible smell. It smells like a cross between rotting cabbage and vomit, and it's just terrible. But it is an incredible molecule because in the gut, when it's produced in the gut, it's actually absorbed by the cells that line your colon. We call them colonocytes, and those colonocytes are very, very important for keeping you healthy. And it turns out that those colonocytes use butyrate for energy, so that's how they live. They live very well. So they're in a symbiotic relationship with us. The microbes are making the butyrate, and we are absorbing that and using that for energy. So it's quite a remarkable symbiosis.
Interesting. So I'm seeing a bunch of questions come in about your lab. So the first one, why don't we start with, how long did it take for you to build this lab?
Oh, well, I've been at the University of Guelph for 11 years now. And when we started out, I just had one little lab and one piece of equipment, really, just one of these systems behind me. And now we have several of these systems, and we have another one coming soon. I just started out with one very small anaerobe chamber, like the one that I showed you. Now we have about 6 or 7 of those. And so I also have 20 people working in my lab. So it grew very fast. It's very busy.
You just answered my second question there, so that's great. Mr. Chasen's class is wondering, do probiotics have an effect on the microbes within your body?
Oh, that's a very nasty question for me, just because it's a very difficult one to answer, because it turns out that not all probiotics are the same. So when you go to the pharmacy and you pick up a probiotic, it's not the same as the next probiotic on the shelf. Like the probiotics are, for those that don't know, they're live microbes that if you eat enough of them, they're supposed to give a beneficial effect on your health. And some good examples of probiotics are the types of microbes that you find growing in certain types of yogurt and yogurty drinks. I don't know if anyone's ever had some of those. Those are probiotics. Now, there's been a lot of controversy in the literature recently because in the scientific literature, it's where we publish all of our results. And that is saying that, that there's a lot of hype about probiotics. And it turns out that a lot of these probiotics that you buy off the shelf don't do very much. Now, some of them, though, do. And that's the difficulty here, because there is no such thing as one probiotic. There's many different types. They all have a different effect on each person. And every single person has a different microbiome. And so a probiotic that works for one person may not work at all for the next person. So it's a very difficult question for me to answer. But I think whether or not they stay, that the other really interesting thing about probiotics is when you take a probiotic, you think that it might colonize your gut, so kind of stay. It actually doesn't. It actually is lost very quickly from the system because there's not enough room for it to take up residence usually, because your gut, if you're healthy, is full of really good microbes that are filling that space already.
Well, that's good to know.
Yeah.
This is a sort of a related question. So when we take antibiotics, when we go to the doctor and get prescribed antibiotics, are we losing bacteria in our body?
Yes, you are. And actually, there's a group of scientists very concerned about that right now, and I'm one of them. Now, I should just say, first of all, that if your doctor gives you or prescribes you an antibiotic, it's very important that you take it, because nowadays doctors understand the dangers of antibiotics, and they've weighed up that equation in their minds, and they've decided that it's actually better for you to take the antibiotic to get rid of an infection. Now, when you take an antibiotic, that antibiotic doesn't just kill the microbe that's causing the infection, because we don't actually have any antibiotics that can do that just target one or two bacteria. So what happens is we get a lot of what we call collateral damage. So so the antibiotics, you take them, they kill off the microbe that's causing the infection to get rid of the infection, that's good. But they also kill off a lot of microbes, and they tend to be the ones that live in the gut that are doing all the work. Now, the good news is that for most people, you'll probably recover from that fairly quickly as soon as you stop taking the antibiotic. But the danger is really what happens when you take lots of antibiotics in a short space of time. That might change your microbiome, and it might not be able to recover. A little bit like trying to clear cut the rainforest in the Amazon. It's very difficult to grow that rainforest back. It takes a lot of time to get that to work. And so we're just still trying to understand how to help that process, because we understand that antibiotics they're an incredible miracle medicine, really. Without antibiotics, we would really not be able to do a lot of the things we can do in medicine. So we need antibiotics, but we also need to try and find a way to prevent some of the damage that these antibiotics might be able to do.
Miss Burton's class is wondering, why are you creating cells instead of purchasing them?
Why are we creating them? I'm not quite sure I follow. So why are we growing them in this?
Growing them perhaps is a better word, yes.
Okay, I see. So I think the reason for that is because there is no shop or store where you can buy these microbes. So a lot of the microbes that we are working with, the ones that I showed you in the RoboGut there, lots of scientists have always said that they've been impossible to culture, impossible to grow. But we can grow them because we are sort of taking notice of how nature grows them, how we grow them in our own colon. So we can't actually purchase them because nobody sells them. In fact, it turns out that a lot of the microbes, people buy them from me because they need them for their experiments and they don't have them. And so they ask me. And often I give them away for free if it's for research. But if it's for like a pharmaceutical company or something, then that helps my lab make a bit of money.
Well, thank you. We're just about to run out of time here, but before we end, I was wondering if I could have you speak briefly on a little bit about your career path and maybe some advice for students considering something similar for themselves.
Sure. So actually, I fell into microbiology completely by accident. Before that, I always knew I wanted to be a scientist, but I was going to be an astrophysicist, and so that's very different from what I do now. But I realized I'm just not very good at math. I'm good enough, but I wasn't good enough to be a physicist. And so biochemistry was what I did at university. And that's studying all of those chemicals and things that those microbes were making. And then after that, so I was actually in London in the UK. That's when I decided to come to Canada because I figured that I really like Canada. I thought this was my opportunity to move, and so I did. And then I started my career in Calgary, and I worked on pathogens. So I worked at that time on a microbe that causes a very severe kind of diarrhea. It's called E. coli 0157. And after I'd worked on the pathogen, I decided that actually perhaps I needed to really consider some of the microbes that aren't pathogens, because everybody studies pathogens and nobody studies the normal microbes. And so that's kind of where I took my independent career, and that's where I am now.
Well, that's all the time we have for today. Thank you, Dr. Vercoe, for taking the time to show us around your lab and teaching us a little bit about microbes. So next week on PIR Live Event, we'll be discussing bullying in our schools with Dr. Tony Volk from Brock University. More information about these webinars and other PIR educational programs are available at pirweb.org. Thanks for tuning in and have a wonderful day.